Hello, and welcome to this Technology Network's webinar, Transforming Viral Vector Manufacturing with Capillary Electrophoresis. I'm Alexander Beadle, Science Writer for Technology Networks, and your host for today. We have a fantastic presentation in store for you, featuring our esteemed speakers Juliette Laigle, Yolande Masson, and Dr. Stephen Locke. Our speakers will be sharing their expertise on capillary electrophoresis in viral vector production, which we're eager to learn more about. But before we dive into the presentation, let me first introduce Juliette, Yolande, and Stephen. Juliette and Yolande are analytical scientists at Iposkesi, where they collaborate with SciEx on implementing analytical methods for controlling RAAV's genome integrity using capillary electrophoresis. Stephen is the Senior Market Development Manager at SciEx. He has over 30 years' experience in analytical chemistry and has presented at nearly 100 international meetings around the world. Following the presentation, we will have a Q&A session where you will be able to ask any questions you may have to our speakers to gain further insights. Feel free to submit your questions at any time during and our team will assist you. Now, without any further ado, I'm delighted to hand over to our speakers who will share their insights and knowledge with us.
Okay, thanks, Alex, for the kind introduction. Today, my job is to introduce this webinar from a technical point of view and discuss some of the work that we've done with Iposkesi. This webinar, as Alex has probably mentioned, is covering the use of capillary electrophoresis in process control of manufacturing viral vectors. And say I did this work together, well, mainly Juliette or Yolanda did this work and I supported them at Iposkesi. So AAVs are adeno-associated viruses, and they're really used as a transport of genomic material in cell and gene therapy. And they're a really common vector used by the industry in developing cell and gene therapy products. AAVs really are quite complicated and they offer several analytical challenges for people developing these products in the industry. They really have different genomic sizes. You often find that not all the viral vectors are full. Some are empty or partially filled with genomic material. And you can also have small-sized impurities present. You tend to find that biopharma scientists often use multiple platforms to analyze multiple CQS or critical quality attributes. And really each platform or technology used has its own limitations. And obviously the industry requires comprehensive AAV analysis to make sure they're safe for consumers or patients to use. And this is time consuming. Now, one of the technologies used in this space is a technology which is produced by SciEx, and that is capillary electrophoresis. And in this project, Iposkesi has used the Biophase 8800 system, which is a multi-capillary system to develop methods to analyze viral vectors in production and in quality control samples. Now, the Biophase system is the latest system from SciEx. It's designed for ease of use. And really it incorporates two different types of detectors. It incorporates UV and laser-induced fluorescence. And it's a change from the previous system, the P800 Plus, is that these are both incorporated in the system and they just switch between detectors by simply selecting in the software. The system is designed and for simple use and to basically reduce downtime. And you'll see, if you ever know about the Biophase system, that it has temperature control sample storage, which enables you to maintain your sample integrity over time. It is also designed and maximized for system operation. And one of the things you can see on the picture of the system at the bottom here is that it is different to the P800 Plus, our historical single capillary system from SciEx, in that the samples are actually placed in 96 well plates on the side. You can see this sort of if I use the mouse, this sort of a blue trap door here. And this really is designed for future-proofing the system for use of robotics in the future. So we've really thought of designing the system for the future. Now, as I mentioned, it's a multi-capillary system. So rather than running one sample at a time, you actually run 8 samples at a time. And this really helps for parallel processing samples and to improve speed of analysis. This is specifically important when you're dealing with process samples, when we may have many to run at one time, which is a bit different for dealing with quality control samples. And this is one of the areas where I wanted ePOSGAZI to do some development work for us and to see if we could actually use this system on process samples. The multi-capillary also helps to improve development time because you can run 8 samples one time. You can really start to think about doing design of experiments and multiplexing and testing different sample prep conditions in simply one go. You'll see the cartridge here is shown on the right-hand side of the slide. And really, this cartridge is temperature controlled or cooled with liquid cooling. So it's a similar sort of principle to the P800+ system. And that's really enables us to maintain accurate migration times across the capillary array. Now, for this system, we offer several different kits, and only 2 of these kits have been used by the team at ePOSGAZI during this project work. But you can see we offer the basic CESDS protein analysis kit, an isoelectric focusing kit, which is to do with charge heterogeneity, as well as a biophase fast glycan kit. This is glycan mapping or N-glycan mapping for protein therapeutics, and the RNA 9000 purity and integrity kit. Of these 2 kits, the main ones used by Yolanda and Juliet will be the biophase CSDS protein kit and the RNA 9000 kit. And moving on to that, the first of these kits, which Juliet will use and discuss using, is the CSDS kit. And this is used to understand capsid protein quality. The purity analysis of AAV viral proteins is important for the quality assurance and safety of viral vector products. You can also use it as a viral titer. So you could use, for example, a viral VP3 or viral protein 3 as an indication of amount of viral particles and therefore the potency of your sample. The CSDS assay is highly sensitive detection because it uses LIF, and it has the ability to analyze low titer samples. Again, this is important to bear in mind because in process development, often the sample concentrations are a lot lower than the quality control sample. And in protein quality assaying, we tend to use the CESDS method or capillary electrophoresis sodium dodecasulfate laser-induced fluorescent method, which is historically the method which has replaced STS-PAGE in labs. So the purposes in collaboration from SciEx's point of view is to investigate the use of CSDS in the analysis of process samples and improve viral protein size estimation. And at that point now, on this is the place where I end my point, and I'll pass on to Juliet.
To begin, thank you, Steve, for the introduction. So to begin with, we wanted to implement the CESDS-LIF method at ePOSGAZI with an RAAV8DS product. For this, we use the method described in an application note from SciEx. With this method, we add a good visualization of the 10 kDa internal standard on the electropherogram, even with a 100-fold dilution. However, we had no visualization of the VP proteins for our samples, whether it is half diluted or pure. It therefore appears necessary to optimize the method for our products. To do this, we carried out new tests still with the same sample, but using a denaturation and labeling method that we found in the literature. In this method, and unlike the one previously tested, the chromium dye, a fluorescent agent used to label proteins, is added to the reaction mix at the same time as all other reagents. A first incubation is then carried out, followed by the addition of water to achieve a sufficient reaction volume. A second incubation at a higher temperature, so 100 degrees, is then performed. This method allows us to obtain an electropherogram in which we had good visualization of the internal standard, but also of the 3 VP proteins, but with a fairly weak signal for the latter. It was therefore necessary to optimize this method. For this optimization, and on the advice of SciEx, with whom we worked closely during all tests, we started by adjusting the incubation times between 5 and 10 minutes and the incubation temperature between 60 and 100 degrees Celsius for the 2 incubation carried out for the denaturation and labeling of VP proteins. We obtained this type of electrophoregram on which we have a good visualization of the 3 VP protein and even of VP3 prime, which correspond to a truncated form of VP3 protein at 90 and 100 degrees Celsius. We then carried out tested on the 2 incubation, testing each at 60 and 90 degrees Celsius. To select the best condition, we calculated the ratio of the area under the peak corresponding to VP3 to the area under the peak corresponding to the internal standard. This ratio allows us to eliminate any impact of the capillary use on the obtained results. Here, this ratio is the best, so the one with the strongest signal and with good replicates when both incubation are performed at 90 degrees Celsius. This incubation was therefore chosen for further use. We then studied the impact of all the regions on the denaturation and labeling of the VP protein, starting with the DTT. For this, we tested 3 volumes of DTT in the reaction mix, 2, 4, and 6 microliter. By analyzing the result based on the ratio of the area of VP3 to the one of the internal standard, we obtained the strongest signals with the 2 and 4 microliter volumes. Following SciEx's recommendation, we selected the 4 microliter volume corresponding to a concentration of 0.04 molar. In the same way, we studied the impact of the volume of the SDS molecular weight sample buffer and the concentration of the chromeodye in order to obtain the best conditions for the denaturation and labeling of VP proteins. This is how we determine the best condition for the denaturation and labeling of our RAAV8 DS and DP products for the CE-SDS leaf method. With this optimized method, we achieved a very good visualization of the 3 VP proteins as well as the VP3 prime protein. This optimization of the method for this serotype was therefore done. The exactly same method was then performed on a RAAV9 DS product. Here, we still had a very good visualization of all VP protein with a slight profile change, as expected and known from orthogonal methods, such as SDS page. Therefore, the optimized condition on RAAV8 also work on RAAV9. This optimized method was also tested on RAAV6 DS samples, and once again, the same results were obtained, and the optimized condition on RAAV8 work on RAAV6 too. Regarding the comparison of the profiles obtained with this method for the 3 serotypes tested here, we can observe that the migration times of the 3 VP protein are the same for RAAV9 and RAAV6. We have a slight shift compared to RAAV8, but this has already been observed with orthogonal methods. Despite this shift, it's important to note that the separation time between each protein is identical for the 3 serotypes, around 1 minute between VP3 and VP2, and 0.9 minutes between VP2 and VP1. Furthermore, in this test, as observed in all the tests performed during this study, the inter-preparation and inter-injection variability was very low, with a RSD consistently below 5%. Regarding the relative ratio of VP3, VP2, and VP1, we can observe that for our products, it's similar between the serotypes. When we compare this ratio to those obtained in SDS page, we can observe a greater difference. One hypothesis explaining this difference is that with CSDS leaf, we work with peaks area, which makes the method more precise compared to SDS stage, where we work with band intensities on gels. Now that we managed to set up an optimized method for studying DP, NDS, RAAV products on different serotypes, we wanted to see if it was possible to quantify the capsid titer of such samples with this method. To achieve this, we perform serial dilution of a known RAAV8 sample to obtain a regression line indicating the ratio of VP3 area to internal standard area as a function of the capsid titer in capsid pyramid. This standard curve was therefore plotted using 10 points, ranging from around 410 to the 10th to 910 to the 13th capsids pyramid. Based on this standard curve, several samples were quantified using the CSDS leaf method optimized. The obtained results were compared to those from two combined orthogonal methods, qPCR, which provide the viral genome titer of samples, and AUC, which indicates the percentage of empty and full capsids. The recovery when comparing these two types of titers is between 68 and 132%, which are acceptable results. After having successfully optimized the method to test and quantify different RAAV serotypes at final steps, so DS and DP, we wanted to see if we could test in-process control sample using the CSDS leaf method. To do this, we tested RAAV8 and RAAV9 samples at early step of the production processes. So here, lysed harvest and unlysed harvest, using the CSDS leaf method optimized for our products. The resulting electrophoregram showed good visualization of the internal standard, but no visualization of the VP protein due to a high background noise. It therefore appeared necessary to optimize the method for the IPCs. Following SCIEX's advice, we tested numerous parameters to obtain better results with the IPC, including changing the sample matrix before protein denaturation and labeling using a column, as well as injection into the biophase using pressure rather than voltage, as it has been done to date. Despite all our attempts, the electrophoregram remained identical with no better visualization of the VP proteins. We then tested a new purification system, and with this one, we obtained electrophoregram with a good visualization of the internal standard, and most importantly, clear visualization of the 3 VP proteins. This new system therefore seemed to allow good purification of RAAV8 IPCs. The same purification system was then tested on RAAV9 IPCs, and the results were identical, with a good visualization of VP proteins. This system therefore also seems to work effectively on RAAV9 IPCs. Okay, so until now, I've told you that we managed to obtain the CSDS leaf method to get a good visualization of the peaks corresponding to the 3 VP proteins. What I haven't talked about is the estimation of the size of these proteins. Here we have 6 RAAV8 DS or DP samples and 1 IPC sample after purification in duplicates. When we use the molecular weight marker supplied by SCIEX, which contains 7 peaks, as shown here, to convert the migration times into size, we systematically obtain sizes greater than the theoretical size of the 3 VP proteins, with a recovery equal or above 130%. We had the same result on leaf and UV detection, so with and without the use of chromeodine. Once again, with the help of SCIEX, we decided to use a ProGen standard containing the 3 VP protein as a new molecular weight marker. This gave us a marker containing 4 peaks, including the 10 kD internal standard, that is structurally much closer to our product. When this marker is used to convert the migration times of the peaks in our sample into size, we obtain sizes very close to the expected ones, with recovery around 100% for the 3 proteins. This protein standard allows us to overcome the limitation of the SciEx molecular weight marker for our products. So now, if we want to conclude on the work done with the CES-DS-LIF method, it's important to note that just because an application node for a method is available, it doesn't mean it will immediately be suitable for your products. In our case, we clearly saw that it was necessary to go through a method redevelopment phase for our viral vectors. This method was then optimized and has proven to work very well on different serotypes, and also allowed the quantification of the capsid present in our samples for the DS and DP products. Moreover, with the addition of a purification step, the method also works on IPCs. More detail on this purification will be available in the application note written by SciEx and Hyposkesi, which should be available before the end of the year. Finally, the size issues encountered with the method were resolved by using a molecular weight marker similar to our products. And just for information, this work is still ongoing, particularly for testing other serotypes.
Thank you, Juliette, for a great summary of the work that you've done at Hyposkesi in Paris. I think it's been, for me, been a really good collaboration, and we've really got to answer some of the questions that I had when we first placed the system in your lab in Paris. Just before we move on to the next speaker from Hyposkesi, I'd just like to go through what Yolanda will be presenting about. We had 2 real areas of interest when we placed the system in Hyposkesi. One was using CSDS for protein profiling, and the other area, which Yolanda will talk about, was using CE for genome integrity and sizing. This again is another really important critical quality attribute for viral vectors, is that you need to understand if your viral particles actually contain genetic material or single-stranded DNA in the case of AAVs. And the quality of this transgene inside the viral vector impacts the infectivity, efficacy, and safety of the gene therapy products. So understanding the impurities or genomic impurities present is also important. The transgene in the genome cassette could be, for example, not present, as you get an empty capsid. It could be truncated, where you're missing part of the genomic material. Or it can actually not present and contain fragments of host cell or plasmids, which are used in the production of viral vectors. So you need to understand if the genome material in your sample is encapsulated, if it's the right size, or if it contains impurities from the production. And one of the methods to do this, again, is using CE. In this case, we use capillary gel electrophoresis, and again, we're using laser-induced fluorescence detection. This is important, again, due to the sensitivity requirements for this particular assay, because as I mentioned with the SDS assay, there's not often the concentration of the active ingredient is quite low. For the purpose of this collaboration, again, we wanted to investigate whether we could use capillary gel electrophoresis for genome integrity of process samples from viral vectors. And we also wanted to look at improving the genomic sizing of the payloads by CGE. Specifically, now we're looking at single-stranded DNA rather than RNA sizing.
Thanks, Steve, for the introduction. For the implementation method of the CGE-LIF, we start working on the extraction step of DNA from our AAV product. We start with the setup of an automated magnetic bead extraction of our DNA using the Kingfisher Flex and a GNase1 treatment to reduce DNA impurities. To obtain the minimum required sample quantity of 1×10⁻11 vg per ml for the analysis, we had to adapt the extraction conditions. Here, we can see the results obtained on AAV8 at different stages of the production process. from lysed harvest to the DS steps. Our peak of interest, framed in yellow, is more or less visible depending on the steps of the production process. Framed in blue, you have also the nucleic acid impurities. At the lysed and chlorived harvest steps, there is still a lot of contaminated nucleic acid, which reduces the intensity of our peak of interest. The steps after chromatography are much cleaner, and our peak really stands out. During this production, we also enriched a part of the product with empty and full capsid. And you can see here the peak of viral genome in the sample enriched with full capsid, and is it absent in the sample enriched for empty capsid. The CG-LIF method allows to visualize the viral genome during the step of production. Then we worked on setting up a standard curve that would allow us to define analysis range for quantification of viral genome. We have tested the two possible injection modes on the biofiles. Electrokinetic or pressure injection give a range of a AAV from 1 to the 10th to 5, 10 to the 12th VG per ml. Using pressure gives a larger linearity range from 3.5, 10 to the 10th to 1.5, 10 to the 12th VG per ml. On the other hand, the electrokinetic injection requires two ranges to be carried out with injection times of 10 or 20 seconds to cover more or less the same range. The choice between electrokinetic or pressure injection is depending on the titer and the step of the production of the sample. During our experiments, we noticed that the size of our peak of interest was always underestimated. We therefore tested with a vector containing a larger genome and compared with an orthogonal method, alkaline gel electrophoresis. This method is less sensitive, no detection of impurity, for example, but gives information on the genome size. Here, you have the electrophoregram obtained on the biofiles and an alkaline agarose gel for ARV8, about 3,000 nucleotides, and ARV9, about 400 nucleotides. The results obtained are shown in this table. We can see that the size of the ARV are always underestimated on the biofiles, whereas a correct size is obtained on the agarose gel. We therefore asked whether this could be due to the use of an RNA ladder to determine the size of ssDNA, or whether DNA was not sufficiently denaturated. Trying to improve the DNA denaturation, we have tested several parameters, such as the temperature and the time of denaturation, increasing separation temperature in the capillary. We have tested the addition of chemical compounds to denature or stabilize the DNA denaturation, but there was been no improvement and even sometimes a deterioration in the signals with a lower intensity. We also tried adding the ladder to a sample in order to be sure that the sample matrix after extraction have no impact on the migration of the samples on the electrophoregram. But in the end, we still have an underestimation of the size. The question is now to determine if the issue in size determination provides from the RNA ladder. We therefore looked for a single-strand DNA ladder to find out whether the underestimation of the size might be due to the use of the RNA ladder when we wanted to determine the size of a DNA. We obtained a commercial second-strand DNA marker. You can see here the different characteristics of the two types of ladders. Here, the SciX RNA ladder contained 8 peaks from 300 to 9,000 nucleotides, whereas the DNA ladder contained 6 peaks from 1,100 nucleotides to 7,200 nucleotides. We used it at a concentration on 90 nanogram per microliter. We compared both ladders. When we determined the size of the DNA ladder using the RNA ladder on 4 capillaries, we found approximately the expected size for the first peaks, but we did find an underestimation of the highest sizes, as shown here by the decrease in recovery as the size of DNA increased. That confirms the underestimation of the higher size DNA with the RNA ladder. When we use this second-strand DNA ladder to determine the size of our 2 AAVs, we found sizes closer to what would be expected in both cases, compared with the values obtained with the RNA ladder. This shows that using a second-strand DNA ladder is better for size determination per our product. The size of the AAV9, about 4,000 nucleotides, is despite everything underestimated, perhaps due to the lack of separation of the peaks of the ladder. We are going to work on the improvement of the peak separation of the ladder to have a better size determination. And now I let Steve to conclude this webinar.
Thanks, Yolanda, for the great summary of the work you've done on genome integrity and genome sizing by CE. At the end of this webinar, I would like to just make some conclusions of the work we've done together at ePOSCASI. I think we started off by asking several questions, and these really, we're getting to the answers of several of these questions now with the work that the team of Juliette and Yolanda and the team of ePOSCASI are doing. The first, and this sort of summarized in the slide. So we've actually seen that a CSDS method for viral protein ratios should be optimized for new sample serotypes. And this is really especially important for looking at sample preparation conditions. I think one thing you have to bear in mind is that different serotypes are denatured differently, so you have to really look at the denaturing step to get the best out of the method. This is why we always recommend you should check and optimize your methods when you're looking at different new serotypes using CSDS. We have seen that Juliette has managed to get a linear response for the optimized assay and produces similar quantitative values and viral protein profiles to orthogonal methods for the different types that she's evaluated. I know she's evaluating more in Paris. And we've also seen that a new sample preparation method enabled the CSDS method to track viral particle concentration and protein ratios in process samples. And this is an important area we want to look at from the position of SciX. And I'm so glad that I said the team of ePOSCASI looked at developing and modifying the sample prep positions for process samples, and which has enabled us now to use this technique to track process samples as well as QC samples. I think the other thing which is really good is what the team has also done is use a different sizing standard to enable us to more accurately measure the viral protein size as well. And by using this ProGen standard, which again we only recently tried the last couple of months, this has really enabled the team at ePOSCASI to accurately measure the size of the viral proteins as well. And this is really an important, I'll say, conclusion, in that if you're going to measure size accurately by CE, it's best to use standards with similar structure to measure the size by CE. And this really shows the importance of choosing the correct ladder when you're doing this type of work. And then I can move on to the work done by Yolanda. And again, this is really important for us to investigate the use of CE, again, on process samples for genome integrity. And again, by using sample preconditions, in this case, mimetic beads with a Kingfisher flex. Yolanda's managed to detect single-stranded DNA in process samples as well. So we can now apply CE to both viral protein profiling and viral titer concentrations in process samples, and genome titer concentration and genome profiling in process samples as well, which is again really important for uptake of this technique earlier in the manufacturing of these particular biopharmaceuticals. And again, the plan is to release more detailed information and application notes in the run-up to the end of the year. And finally, I think this is an important area that I personally wanted to test as well, is to improve the sizing, specifically of single-stranded DNA in this AAV viral vector. We've historically at SCIEX, we've managed to get good sizing when we've compared and looked at RNA genome material with an RNA ladder, but we always found that when we used the RNA ladder for single-stranded DNA, our sizing wasn't very accurate. And by actually moving to a single-stranded DNA ladder instead of an RNA ladder, we've really improved the accuracy of our sizing as well. We're not quite there yet. As Yolanda mentioned, we still need to investigate several questions we have in this space, and this is an ongoing project with ePOSCASI to look at this area in the up and coming months. At the end, I'd like to acknowledge some people who have helped us do this project together. I think for me, this has been one of the best collaborations I've ever run, and I really want to thank the team at ePOSCASI working with SCIEX to help answer some of these questions which the industry wants the answers to. But also, I don't work alone. I work with a great team within SCIEX, and I'd like to specifically acknowledge two of my colleagues, Marcus Heinel. Marcus Heinel has been brilliant in helping me answer some of the technical questions and supporting ePOSCASI in the method development. Jean-Charles Belay is helps me with supporting ePOSCASI with consumables and discussions to help the project move forward. And really, I need to again thank the team at ePOSCASI. You've listened to Yolanda and Juliette, who are some of the key researchers involved in this testing, but there's also a big team at ePOSCASI as well, which have been supporting this work, including Nadia, Corotine, Litia, and Oriore. And again, a big thank you to the team at ePOSCASI. Without their support, this work would not have happened so well. And again, thanks everyone for your attention today. And now we can open up to questions that you may have on this work.
Thank you to our speakers, and thank you all for your active participation. I hope you found this presentation as insightful and thought-provoking as I have. Before we start the Q&A session, we would appreciate your responses to a couple of short polls that should now be on screen. Please select the answers that are most relevant to you. Thank you for your responses. We will now begin the Q&A session, where you have the opportunity to ask your questions to the experts. To submit your questions, please use the chat box on the right-hand side of your screen. Type your question and click send. We will do our best to answer as many questions as time allows today. Now, we've already received several questions while the webinar was underway, so I'll begin with those for now. And the first question I have, and feel free to jump in, whoever wants to take this one, is what was the effect of using a different protein ladder for sizing?
Hello, I will take this question. So as I shown during the presentation, when you use a Progen standard as a molecular weight, you have a better sizing of our viral protein for our product, because the the standard is quite more similar to our product. So it helps us to have a good sizing of the proteins.
That's fantastic. Thank you. The next question that I have for you here, what was the lowest genome concentration you detected in your AAV process samples?
The lowest concentration is 2.5 10 to the 10th with an electrokinetic injection, but this concentration is outside of the range of linearity.
That's great. Thank you. Moving on, we have this question. How easy is the sample cleanup process? How easy is the sample cleanup for process samples?
It's really easy. You will have more information of this cleanup in the application note that will be available by the end of the year. But I can tell you that it's quite easy.
Fantastic. Thank you. We also have this question. How many different serotypes of AAV have you tested?
Around 5 serotype. We have done AAV8, AAV9, AAV6, and some AAV2 and AAV5. And I show all the results in the presentation. Maybe not for the AAV2 and AAV5, but it will be available in the application notes.
Thank you. How does the analysis of lentivirus versus AAV change when you do genome integrity?
We are still working on the lentivirus's genome integrity. The process on the biofiles is similar, but the preparation of the sample is different.
There's also the accuracy of the genome sizing is different, isn't it, Yolanda? Yeah. So the RNA ladder works better with lentivirus than it does with the single-stranded DNA and AAV.
That's great. Thank you. Moving on, we'll take this question. Are the kits GMP grade?
Yeah, they're manufactured for QC, so they are GMP grade. We manufacture them all at SciEx, which are the kits that Yolanda and Juliette have used.
Fantastic, thank you. And here, did you look to optimize incubation temperatures for the other serotypes, or just stick with the fixed temperature established for AAV8?
We test the temperature that we optimize on AAV8 on other serotypes. And because we saw that the result was really good, we didn't optimize the temperature. But just for information, we have a platform process to produce our samples. So maybe it's why we have the same response on the optimized temperature for the labeling and the denaturation of the protein.
Fantastic, thank you. We also have this question here. Have you evaluated MW modification after labeling, brackets, covalent labeling, for each protein?
Yes, we saw that. So it's a covalent labeling.
Yes.
And we saw that when we use the chromeo or we don't use the chromeo, we have the same size for the molecular weight. So it's with the overestimation of the size is not due to the chromeo dye.
That makes sense. Thank you very much. Moving on, we'll pick another question here. Are the analytical methods qualified and validated?
Not yet. But now that we finished the development of the method, we are going to do the qualification and the validation of the CESDS leaf method. It's ongoing.
Fantastic. Thank you very much. And here, We discovered that the 10 kilodalton standard has a small level of impurities at higher molecular weights. If you spike the sample with the 10 kilodalton, how can you measure the purity of the drug product or drug substance?
Yes, so during our tested, we didn't see this type of impurities for the 10 kDa. So I can't answer this question because we don't have this impurity for our with our optimized method and conditions.
If you tend to get an impurity with an internal standard, you tend to reduce the concentration of the internal standard to reduce the effect of the impurity. That's something we've done before. We've seen impurities, but we never saw it, did we, Juliet, in our work as in
No, but it's quite dilute in our
yeah.
in our process. So maybe that's why we don't have any impurities. We don't saw them.
That makes sense. Thank you very much. And I think we have time for one final question here. Each drug product is probably formulated in different buffers. Have you pre-treated the samples before labeling to avoid matrix interference?
So at Hyposcasy, we just have 2 formulation buffer, and we don't see any impact of the formulation buffer on the method, on the results. So we don't have some treatment before the labeling.
Fantastic. Thank you. Unfortunately, that is all the time that we have for today. For any questions that we couldn't address during this session, rest assured that we will respond to them offline as soon as possible. You can also continue to ask questions when watching this webinar on demand, and these will be sent over to our speakers offline. Finally, don't forget to download your certificate of attendance from the Handouts tab on the right-hand side of the webinar platform. Once again, thank you, Juliet, Yolanda, and Stephen, for your time today, and thank you, everyone, for watching.
Thank you.
Thanks, everyone.